Hi viewers, in this class we are going to discuss the classification of antibodies and the diversity of antibodies, otherwise types of immunoglobulins and the genetic basis of the diversity of the immunoglobulins. Under this topic we are going to discuss introduction to the topic, what is the basis for the classification of antibodies, different kinds of antibodies like IgG, IgA, IgM, IgD and IgE, and finally the genetic basis of antibody diversity. There are 5 different types or classes of antibodies are recognized. The distinction of all these 5 based on the difference in the constant region of the antibody or Fc region of the antibody. Fc region of the antibodies that contain 5 different kinds of heavy chains. All these antibodies are further divided into subclasses based on the heavy chains that they possess. The heavy chain differs as an antigen in carbohydrate content and in size. Most importantly, they confer different biological functions on each isotype. The heavy chains derived from the various immunoglobulin isotypes are designed with the Greek letters. Let us see immunoglobulin G that has gamma type of heavy chain. Immunoglobulin A, alpha type of heavy chain. Immunoglobulin M, mu type of heavy chain. Immunoglobulin D, delta type of heavy chain, whereas the immunoglobulin E that contain epsilon type of heavy chain. So there are 5 different classes of antibody molecules are there which contain different kinds of heavy chains in their structure. Let us see one by one. Here you can observe this is the gamma type of heavy chain and this is the alpha type of heavy chain, a mu type of heavy chain and the delta type of heavy chain, and finally this is the epsilon type of heavy chain. Thereby they have given the names like IgG, IgA, IgM, IgD and IgE. So that is constant region of the heavy chain that determines the isotype of the antibody that we should remember. Let us start with the immunoglobulin G. Immunoglobulin G, IgG. IgG is the predominant immunoglobulin in blood, lymph fluid, cerebrospinal fluid (CSF) and the peritoneal fluid. IgG is an abundant class of antibody, particularly in the human serum. Its amount is of about 50% in the total proteins. 50% in the total proteins. And the IgG molecular weight is approximately 1,50,000 daltons. This IgG is produced in the body unless there is a secondary response. This antibody diffuses through wall of the blood vessel very easily than the other class of antibodies. Blood capillaries are lined by the endothelial cells because the endothelial cells are very simple in structure but very complicated in biological functions. They show selective permeability, that is, they allow certain material to come inside. Then the basic property of the endothelial cell is when there is a contact of the antibody or macrophage or a solid particle, the adjacent cells give away to antibody by moving towards opposite directions to each other. IgG found majorly in the tissues These immunoglobulins diffuse across the placenta. They pass through from the colostrum, the first mother milk. It plays a vital role in passive immunization for the newborn against the infections. These molecule has the ability to bind Fc receptors and phagocytic cells and activate the complement system. This Ig has the greatest half-life period, that is of about 23 days. They participate in opsonization, antibody dependent cell mediated cytotoxicity, and also in the neutralization of the bacteria and the viruses. These are the properties and the functions of the IgG molecule. It is a monomer in structure. It is a monomer in structure, that we should remember. This is all about the IgG molecule. See, here we can observe, here it is the hinge region of the antibody. These are the antigen binding sites, FAB region, and this is the FC region of the antibody. So this is a generalized antibody structure. Coming to the immunoglobulin A or IgA. IgA is the major immunoglobulin in external secretions such as saliva, mucus, sweat, gastric fluid, and the tears. IgA found predominantly in the mucus. It is moreover the major immunoglobulin of colostrum and milk, and it may provide the neonate with a major source of intestinal protection against the pathogens. The molecular weight of a monomeric IgA approximately 1,65,000 daltons. 1,65,000 daltons. During the synthesis of IgA by plasma cells situated in various epithelia of the body, two monomeric IgA molecules become joined by a J chain. There are two monomers. This is one monomer of IgA. This is another monomer of IgA. Each one has 1,65,000 daltons molecular weight. So 1,65,000 and 1,65,000 daltons. So both of them, they are connected through a joining chain, which is nothing but the J chain. So two monomeric IgA molecules become joined by a J chain. The J chain synthesized by the same cell that makes the IgA is attached to the IgA molecules by the disulfide bonds. That is joined together by the disulfide bonds. So whenever there are the joining of two isomers, it is called as the dimer. Previously we have discussed the IgG, that is the monomer, only single IgG molecule. But here two IgA molecules fused together, combined together through the J chain, through the disulfide bonds. So hence it is known as IgA dimer. This IgA dimer released into the lamina propria and transported across the epithelial cells of the mucosa into the lumen. This transport is mediated by another protein called the secretory component, which is a receptor on epithelial cells that becomes attached to the dimer of IgA. The IgA found in secretions is always present in the dimeric form and has a molecular weight of about 4 lakh daltons, means 165, that is 3,30,000 daltons, and the secretory protein, it is of about 70,000 daltons. Totally it is 4 lakh daltons totally. That is two IgA molecules, two monomers of the IgA plus secretory protein, that is 70,000 daltons. And what is the use of the dimer is, the dimer enhances the binding site and remain for a longer time. The IgA present in the serum is predominantly monomeric, has been released before dimerization so that fails to bind to the secretory component. Secretory IgA is very important biologically, but little is known of any function of the serum IgA. IgA play a major role in defending the mucosal infections. It also shows bactericidal activity and it also shows antiviral activity. This is all about the dimer of the immunoglobulin J, otherwise IgA molecule. So here we will see the immunoglobulin dimerization through the J chain. See here, this is IgA and this is one more IgA. This is the J chain and the orange color one is the secretory component or secretory protein. So 165,000 Daltons, 165,000 Daltons and the 70,000 Daltons, totally 4 lakh Daltons. See here, these are the two monomers, J chain. So it is a dimer where it is coupled with the secretory component or secretory protein. So it is a monomer and it is a dimer along with the secretory IgA. Coming to the third type of immunoglobulins, that is IgM. IgM is the first immunoglobulin produced following the immunization. That is, whenever the antigen entered into our body, the first kind of antibody which is produced against the infectious agents is the IgM. Its name derives from its initial description as the macroglobulin, large globulin, because of its heavy molecular weight, which is of about 9 lakh Daltons. The IgM molecule consists of 5 monomeric units. So IgG only monomer, IgA is dimer, but the IgM is the pentamer. Penta means 5 single units are there. So let us see 1, 2, 3, 4, 5. The IgM molecule consists of 5 monomeric units. Each of it consists of 2 light and 2 heavy chains, all joined together by additional disulfide bonds between their Fc regions and by a polypeptide chain termed as the J chain. So all of them bound together at their Fc portions. So these are the Fc portion and these are the FAB portions. So at the Fc portion, all of them joined together through the J chain. The J chain synthesized by plasma cells has a molecular weight of about 15,000 Daltons. 15,000 Daltons. The IgM appears in pentameric structure. The IgM present in the adult human serum is found predominantly in the intravascular spaces. IgM is also found on the surface of mature B cells where it serves as the specific receptor for the antigen. Only this class of Igs, this class of immunoglobulins that is synthesized by the fetus beginning at approximately 5 months of gestation. At the age of 5 months, when the baby is in the womb of the mother. Elevated levels of IgM in the fetus are indicative of congenital or perinatal infection. So, in general, there is no infection when the baby is inside the mother's womb, but sometimes there is every possibility to get the infection. Thereby, that is indicated by if the developing child, if it gets infected, that is shown by the elevated levels of the IgM molecules. Surprisingly, each pentameric IgM molecule appears to have a valency of 5 instead of the expected valence of 10 due to the 10 FAB regions. Any large antigen bound to one of the FAB regions may block a neighboring site from binding with antigen, making the molecule appear to be pentavalent. Let us see here, if these are the 5 antibody structures bound together, and valency of each antibody, that is, what is the valency? So the ability of the antibody to attach with number of antigens. So one antibody can bind to two antigens at a time. So this is the valency of the antibody. So the valency of any monomeric antibody is only 2. So there are 5 antibodies, hence the valency, maybe expected valency is 10. So that is one pentameric IgM molecule has the ability to bind 10 antigens at a time theoretically. But what happens practically? For example, this is a first antibody, this is the second antibody, let us assume. If any antigen which is very large, if it is bound to the first antibody with its epitope, it may block the another antibody's FAB portion. Hence what happens? The valency of the antibody decreased. So due to the overlapping of the antigen for the adjacent antibody's FAB regions, the pentameric antibody unable to attach to 10 antigens. So it is only theoretical expected valency. So in general it may attach to 5 antigens or 6 antigens at a time. So that is, the theoretical valency is 10, but it may fail to attach that many number of antigens at a time due to the large sized antigens which are attached to one FAB region of one antibody. They may overlap the FAB region of the another antibody, and thereby they block the attachment of the antigen. So that the practical valency is only 5. So here these are the disulfide bonds and this is a J chain. So the heavy chain is mu type, light chains are different, kappa or lambda. And coming to the fourth kind of antibody, that is immunoglobulin D. Immunoglobulin D found to be a major surface component of many B lymphocytes. Immunoglobulin D in serum is very low in quantity. On B cells, it serves as a marker of the differentiation of the B cells to a more mature form, but its exact function as a receptor for triggering or differentiation is still completely not clear. And moreover, it is a monomer. IgD is present in the form of a monomer with the molecular weight of 1,80,000 Daltons. According to the latest theories, they are fundamentally involved in the maintenance of the B cell tolerance. Maintenance of the B cell tolerance. This is about the immunoglobulin D. See, here it is a monomeric form, IgD. And the last kind of immunoglobulin is the immunoglobulin E, where it possesses the epsilon kind of heavy chain. IgE is also called as a reagenic antibody, is present in the serum in the lowest concentration of all the immunoglobulins. IgE has a molecular weight approximately of about 2 lakh Daltons. The heavy chain of IgE contains an extra domain by which it attaches with unusually high affinity to specific receptors on the mast cells and the basophils, where the IgE molecules may remain for weeks or months. It has a role in protection against microorganisms. Elevated levels of IgE in serum have been shown to occur during infections with certain parasites. Immunization with Ascaris antigen induces the formation of IgE. The surface of highly granulated basophils or mast cells contain receptors for the IgE, and IgE predominantly attached to these cells. When an antigen binds with the IgE attached to these cells, the cells become activated and release contents of their granules. These substances are called as biologically active substances or pharmacologically active substances in the form of histamine, heparin, leukotrienes, serotonin, prostaglandins, and other active compounds trigger the hypersensitivity reactions. So, the IgE is called as the inflammatory mediated antibody. Inflammatory mediated antibody. So that is the IgE play a major role in the anaphylactic reactions, otherwise type 1 hypersensitivity reactions, otherwise allergy reactions. Here there is an overview of all the antibodies: IgG monomeric form, IgA dimeric form, IgM pentameric form, IgD monomeric, IgE monomeric. So here IgG gamma type of heavy chains, A alpha type of heavy chains, M mu type of heavy chains, D delta type of heavy chains, and E epsilon type of heavy chain they possess. Here this is a class of the immunoglobulin, main structure and the function in the circulation, main structure in the mucosa and function in the mucosa. So here antibody, their functions and the locations of the antibody, whether that is membrane bound, otherwise it is secretory. And coming to the genetic basis of antibody diversity. What is the basis behind the differences among all these antibodies? One characteristic feature of the immune response is its enormous diversity. Estimates of the number of B and T cells with the different antigenic specificities in a given individual range from 10 to the power of 6 to 10 to the power of 8. If every immunoglobulin or T cell receptor were coded for by one gene, then an individual would have to have the same number of genes devoted exclusively to coding for these structures. Since such a large number of genes would occupy a significant percentage of the individual's genome, that is inherited DNA, we know that genes coding for immunoglobulin and the T cell receptors use a unique strategy to achieve the degree of diversity required. This strategy uses a much more limited set of genes, numbering in the thousands rather than millions. The first key finding was that the variable and constant regions of an immunoglobulin molecule were coded by different genes. In fact, many different variable region genes can be linked up to a single constant region gene. The combining of variable and constant region genes considerably reduces the amount of genetic information required to encode different antibody molecule. A subsequent finding by Susumu Tonigawa was the Nobel laureate. He found that antibody genes could move and rearrange themselves. A subsequent finding by the Susumu Tonigawa was that antibody genes could move and rearrange themselves within the genome of a differentiating cell. For this finding, he was awarded Nobel Prize in the year 1987. Here you can observe this is the DNA of the individual, and here there is a rearranged DNA. So this is for variable region and this is for constant region of the heavy chain, and here variable region and constant region of the heavy chain. See this is the light chain and this is the heavy chain. So this is the germline DNA, natural DNA. This is the rearranged DNA. From this DNA there is every possibility of the transcription of the mRNA that undergo translation to form the antibodies, heavy chains, light chains, especially the variable light chain, variable heavy chain, constant light chain, and the constant heavy chain. A variable region gene can be located in one position in the DNA of an inherited chromosome, that is the germline, and can then move to another position on the chromosome during lymphocyte differentiation. This process of rearrangement during the differentiation brings together an appropriate set of genes for the variable and constant regions. This set of genes is then transcribed and translated to complete the heavy chains and the light chains. Every individual synthesizes an enormous number of different immunoglobulin molecules, each of which can act as a receptor on the B cell surface specific for a particular epitope. The variable region of heavy chain immunoglobulin molecule is coded for the three separate genes referred to as VH region, variable heavy chain region, and DH, D for diversity, V for variability, and D for diversity, and J for joining the gene fragments. So the variable region of heavy chain of the Ig molecule is coded for by three separate genes referred to as VH, H for heavy chain, variability in the heavy chain, diversity in the heavy chain, and joining of all these small gene fragments. A distinct gene segment codes for the constant region of the heavy chain, that is CH. The variable region of a light chain immunoglobulin molecule is coded for by two gene segments which are called as VL and the JL. Variability of light chain and joining of the light chain, distinct from the gene segment used for the heavy chain synthesis. Every cell in the body, that is in the germline, contains multiple VDJ gene segments, VDJ gene segments for immunoglobulin H and L chain, heavy chain, and the light chain synthesis. In the course of differentiation, a B cell rearranges its heavy chain DNA so as to join one VH gene segment to one DH gene segment and one JH gene segment. The joined VDJ unit codes for the entire variable region of the heavy chain. These gene rearrangements put the VDJ unit next to the heavy chain constant region genes, that is the constant heavy chain mu or constant heavy chain delta, that is IgM and IgD. These are the primitive antibodies which are present on the surface of the mature B lymphocytes. The same type of rearrangement occurs to produce a gene unit coding for the entire V region of the immunoglobulin light chain. One VL gene segment is joined to one JL gene segment. This VJ unit is put next to a light chain constant region. When DNA rearrangement has occurred, the antigenic specificity of that particular cell is fixed. Primary DNA transcripts are made from the rearranged DNA. Unnecessary RNA spliced out of the primary transcripts, resulting in mRNA for light and heavy chains, which are then translated into L chain and the heavy chains of the IgG and IgD. So this is the light chain and this is the heavy chain. This is the variable light chain and this is the constant light chain. Here variable heavy chain, constant heavy chain, one region, two region, and the three regions. After antigenic stimulation, a B cell can further rearrange its DNA. The VDJ unit, which has joined to the C. Constant mu chain and constant gamma genes can rearrange to join another constant region gene such as C gamma, C alpha, C epsilon. This phenomena is known as the class switching. As a result, the B cell that was synthesizing IgM and IgD can now synthesize antibodies of different isotypes, that is IgG, IgA and IgE, based upon the need of individual. So which kind of antigen is killed by which kind of antibody, that is made possible after the antigenic stimulation, which is called as the class switching. Then diversity in the antibody specificity is achieved by four factors. Number one is multiple inherited genes for the V regions of both light chains and heavy chains. Second one, rearrangement of V, J and D gene segments in different combinations and random assortment of heavy and light chains. Third one, junctional and insertional diversity when V, D and J genes are joined. And fourth one, somatic mutation, which primarily occurs after stimulation by antigen, leading to selection for mutations that endow the antibody with higher affinity for the antigen. Thus, these mechanisms allow a small number of genes to generate a vast number of antibody molecules with different antigenic specificities. So, this is all about the types of antibodies, that is the classification of antibodies and the genetic basis of the antibody diversity. Thank you one and all.